Let's start our next session on new approach methodologies. So we have got three presentations, 15 minutes each. The first one is going to be provided by Maria Cristina Albertini from the University, from the Department of Biomolecular Sciences, University of Urbino, Carlo, in Italy. And she's going to be talking about a near in vivo microfluidic system to analyze chemicals and nanomaterials, metabolomic responses. Thanks, Andrea, for helping to pull it up, the presentation.
Thank you.
The page is yours.
Yes, I come from a little city, Urbino, and the university is Carlo Bo, University of Urbino, Carlo Bo. This talk is related to CheMatSustain project. So I'm part as partner to this project, where you can see that there are a lot of topics that are in progress. Especially, we are working on chemicals and nanomaterials, trying to use new methodologies for risk assessment. More specifically, what I wanted to talk to you today is about the in vitro and near in vivo systems that we are using as microfluidic systems. I would like first to say that in this case we are directly collaborating with Poland. For example, the University of Lodz, ULodz, is the university, the partner, who is synthesizing the particles, the chemicals and particles, characterize them, and also send to all of the partners the materials to be sure that we are using the same material in our experiments. Then, more specifically, for the in vitro studies, we are using the collaboration with the two universities, Technical University of Lodz. Professor Bogdan Walkowiak is the leader, with whom we are working in the in vitro tests. We also have the support of Dr. Raffaella Corvi. She is from the JRC, the European Reference Laboratory for Alternatives to Animal Testing. This is important because they promote and facilitate the use of non-animal methods in research. This is why I wanted to talk about a microfluidic system, a new methodology that we are setting up, because we are close to in vivo conditions, but using in vitro ones. Of course, in vitro are really important because we can see a direct response of the materials we are analyzing. But if we also add the physiological condition, we may be close to in vivo conditions. In this way, if it is necessary, we can continue with in vivo analysis. In our model, we wanted to mimic blood flow using a microfluidic system. I'm going to talk about the classical one, may I call it classical, in the flask as a static one. In our case, we use, thanks to Ivitech, the company who made this instrument. I can tell you that when you have an instrument or you buy a kind of this instrument, you are collaborating with the company because they have to follow up, not only giving you the instrument, but also following you with your setup. In this case, if you want to mimic blood circulation, you have to consider to mimic the shear stress of the human circulatory system and to study the blood flow and cell behavior in a controlled environment. You can study, in our case, for example, we use cytotoxicity or inflammation response or other metabolic conditions that can be analyzed in this system. The shear stress is something related to physics. is the force per unit area that is created when a tangential force, like the blood flow, acts on a surface endothelium. In this case, you have to consider that the endothelium is really sensitive to hemodynamic shear stress. And this is really important in physiological condition, because in this case, you can maintain the endothelial quiescent cell, you cannot see it here, and their integrity, because endothelium cells are really sensitive to different processes. You don't have to consider them only as part of a structure, of a vessel structure, but also part of some processes, like, for example, inflammation. They can be activated and participate in the inflammation process. In our case, we used this system where you can see multi-DIN, which is formed by 4 chambers. If you want to use such a kind of instrument, you need first to know what you want, which is the aim of your study. In our case, as in the CheMatSustain project, we needed to have a certain number of cells. We need a type of cell, which is the endothelial one, but in this case, we use this kind of cells because they are not primary, they are not tumor cells, but they are hybrid. This is really important because when you have endothelial primary cells like UVEX, umbilical vein, human umbilical vein cells, they can go to senescence after different replications steps. This is called replicative senescence. In this case, we can postpone the replicative senescence and have more time to perform our experiments. This is why we decided to use this kind of cells. Also, when you use this multi-DIN system, these chambers can allow you also to make some microscope analysis, the morphology. You can use also fluorescence microscope, so you can use other types of markers depending on what you need. Certainly, if you want to analyze cytotoxicity of materials, nano or chemicals, you may use different kind of tests. We use, for example, the trypan blue test after detaching the cells from the chambers. Consider that when we work in this condition, you also have to consider the time of treatment. In our case, it lasts for 24 hours, so to consider the whole experiment, we need to take into account that we need at least 1 week, because we need 4 consecutive days to perform the experiment, and then we have the last day dedicated to the washing of the instrument and also to sterilize everything and start again the next week. It was really important in our CheMatSustain project to be really well integrated with the activities of Toul University. The static condition was the first step. Then we wanted to reproduce exactly the same treatment conditions in the microfluidic system. Same cells coming from the same batch, same medium, same time of treatments, so that we could integrate also the analysis to perform. This is why you can see here in the picture, we have 2 colleagues from Toul University in Urbino. The others are my precious colleagues, Serena Benedetti and Noemi. Noemi is a PhD student. She will go to Poland to perform other experiments. So they did the first treatments. We know which are the concentrations to use. We make both cytotoxicity. Then we store the cells From these cells, we are going to do molecular analysis. We are going to perform this kind of inflammation analysis, taking into account the toll-like receptor signaling pathway, for example, where there is IRAK1 able to activate NFKB, which is responsible for pro-inflammatory cytokine production, like, for example, interleukin-6. But it also activates the expression of microRNA, in this case, microRNA 146A, targeting IRAK1. You know that microRNA are non-coding small microRNA that can silence gene expression through targeting the messenger. So analyzing IRAK1, interleukin-6, and microRNA 146A, we can consider this signaling pathway if it is activated or not. At the end, in our condition, as you can see, the first day, you have to consider to seed your cells, the number that you need. You have to consider that after the third day, when it is the treatment day, you need the number of cells that you can use for the analysis. So the first day, there is the coating day with fibronectin, for example. We seed the cells, then we have to wait that they are attached in the chambers. So the day after, we start with the conditioning condition, because we need the shear stress. We need the cells, that the cells are quiescent and have their integrity. This can be performed thanks to the flow at 250 microliter per minute. This condition lasts for 24 hours. And then we can start the treatments, and in our case, it's 24 hours. If you have only 1 hour of treatment, of course, you have one less day to work. At the end, you have to detach the cells, then make cell viability, as we do, to see a first cytotoxicity analysis. Before detaching the cells, you can also take some pictures, because sometimes the cells are alive, but the morphology is different. This is why metabolic response may be different. At the end, there is the washing day. We can define it as a washing day. So we have to detach everything. We have to wash with ethanol 70%, for example, also using UV, and be ready for the next week. So, in conclusion, what I wanted to say is that the new methodology is not only the use of an instrument, but it's also the setup of all the technique and the experimental condition that you need to test cytotoxicity. And in this case, we really thank IVTech company, because they are collaborating as a scientist. I really thank Toul University. We spent a lot of nice days, not only with the hard work. And I would like also to thank our coordinators from the University of Hamburg, the Applied University of Hamburg, especially Jelena Barbier, who is following us every day and making a lot of efforts. And thank you to all of you for your attention.
Thanks a lot, Maria, Cristina, also for keeping the time. We are running a bit late. We will collect the questions also for your talk and the next talk,
okay.
And then we can discuss. So I'm calling Israel. We have colleagues who are not able to travel, and they are going to be giving their presentation as an exception online, but we understand the reasons that Israeli people might have in regard to traveling. So I'm trying to call Dror, Cohen, and Ze'evi, Maor, and sorry for my maybe misspelling of your names, who have been joining via Zoom.
Can you hear us?
Yes, we hear you already. And now, after the endothelial cell model, new approach methodology, we are looking into an ex vivo model for cosmetic testing on skin irritation, sensitization, allergic reactions. Stage is yours. Thanks for joining.
Thank you very much for inviting us and for the opportunity to enjoy the spirit of Venice via the internet. We are happy to share what we are doing in our project and especially how we want to introduce SSbD concept in the cosmetics. Actually, SSbD put a triple challenge. Safety is above all, for sure, and sustainability is also needed today. But of course, no harm to air, soil, or water, and it should be by design, which practically means that all the expenses should be done before the technology is already on the shelf on large scale. So going directly to the cosmetic, there is a very important factor called NOAEL, which is no observed adverse effect, and it is defined in milligram per kilogram and related to the chemical ingredient specific toxicity. Actually, this factor, together with another factor, SED, helps the safety assessor to decide whether the product is safe or not. If the margin of safety is above 100, then product is safe. We are looking for ex vivo promising method to replace the current method, because NOAEL currently was decided following animal testing and which currently is not allowed by the European regulation. So this is a reason that there is a need for other technology, and we want to share with you our ex vivo models that we believe could be a good replacement to the animal test that were existing. Dror will describe you some of our laboratory methods and results.
First of all, hi everyone, and thank you. It's my honor to present here, so thank you for inviting us. I wanted to have some glimpse about what we're doing. Also, it's regarding to the SSbD Chem4 project that we are working at, and what is the model of the ex vivo skin is applying and the advantage in this project about SSbD regulation. As a glimpse, there is, as you saw, there is many kind of different models that can be used for lung, for intestinal, for even for skin. However, in our lab, we are using ex vivo skin models. There is some debate about regarding to the ex vivo, because the ex vivo is actually taken from donors, human donors, from plastic reduction, abdominal reduction, elective one. That is actually we use it from women aged from 23 till about 65, and we take them and as you can see here from the picture, this is how it looked like after being treated and being put in the vial. There is some debate regarding to the reproducibility of this model, and in SSbD, what we had to do was actually to take this model and to take it to the next level as a validation, to show that actually it doesn't really matter which kind of donor you take, we eventually going to get some pool or the same results from any kind of ex vivo skin model. So in general, what we actually tried to do in this project was to take the human skin, as we saw before, and to apply it topically in this project was nanocellulose. in order to test it, we have 3 different models that we decided to choose. It was not just to take the model itself, was also to take this human skin and also apply on it topically SLS, sodium lauryl sulfate, which cause irritation and cause kind of irritant contact dermatitis. We applied also UVB radiation on it, so we have some UVB damage on it. And then the third one was actually to put a nitrobenzene molecule on topically on the skin, and that caused a kind of allergic contact dermatitis. So those 3 models was not just tested for as a human skin model, but also was taken in order to put, when we apply any kind of nanoparticle or any kind of new substance on it, we see that even if we're going to put it on a naive skin, if we see the safety of it, we also want to put it on a damaged skin that was proceed some stressor, and we see that if there is some an effect on it. So in general, those 3 models, we test several kind of biomarkers. 2 of them was the apoptosis activity and viability. The cells we test the same, the cells, and also we test different kind of cytokines that was secreted on each one of the models. So in glimpse, in kind of an eye view, bird eye view on what we're going to show you, actually it's going to be published soon, so we're not going to give details and parameters, but you can see here that if we can see, this is on the left, there is all those kind of parameters which are the cytokines. This is like the levels of the untreated, green regarding to low level, red, yellowish regarding to high level. The red is the highest level. If we apply the SDS, the sodium lauryl sulfate, we can see that there is an increase of many kind of cytokines. If we see the NBB, which kind of the allergic contact dermatitis, there is a different pattern and a different levels of each one of cytokines. And as well, if you see in the UVB radiation, there is a different pattern. What I want to understand that actually, and this is like taken from 5 different donors, the levels of each cytokines was probably pretty much the same. So it was continuing, it was a reproducibility of it. And we can see that each kind of cytokine have its own like orchestra of when it started and when it's end on each kind of the model. So in a glimpse, what we did actually in the project, we took a nano cellulose. Here we can see how the samples look like. On the right, there is the naive skin that was not treated topically by the nanocellulose, but each one of the level was put the stressor. The stressor doesn't have any color, so this is why the skin look naive and without treatment. And on the left, we can see there is some white, on the right, we can see there is some white dot, which is actually the paste of the nanocellulose that was put on top of it. And we test all those parameters for 24, 48, 72, and 96 hours after. And what we can see here, let's see in general, this is again some very from high perspective view. When we test on the top, it's the same result that I showed you before, but on the bottom is actually the result, what happened to the skin that was treated with the nanocellulose. So we see the untreated was kind of the same. We see the same kind of pattern. However, if we see the difference between when we put SDS on the skin, we can see that there is some reduction in cytokine secretion. You can see the same thing happens with the NBB, with the allergic contact dermatitis, and also the UVB shows some reduction in the total inflammation. So we kind of assume, actually it was not such a surprise, because we were assuming that the nanocellulose can actually absorb some of those stressors. So this is why it was reduced by the cytokine levels secreted to the medium. So in general, what we want to kind of show, and what we'll be showing in our going to be published a paper that this ex vivo skin model is demonstrated actually it's very robust and very reliable skin, human skin tool that can be tested for many kind of cosmetic or new material, and as well for pharmaceutical in the future, instead of making an experiment on animals. So it would be quite a very good and easy way to start an experiment. And regarding to each one of the stressor, we can show that each one of the stressor show a specific kind of fingertip kind of a secretion to the medium, and different stressor activate distinct cytokine set. So you can see that some stressor activate I1 alpha, the other activate TNF alpha, and it's each one of them activate a different one. And the ex vivo response mirror kind of the response that actually we see in the human skin in the clinical form. So I'll give to Ze'evi here to say the conclusion for everything we kind of say.
So actually, what we believe that our results is quite convincing that this ex vivo human skin system is a real, reliable, scientifically validated for advancing cosmetic, dermatological, and pharmaceutical product development, which will ensure good predictability, regulatory compliance, and public safety. We want to propose that the margin of safety could be evaluated if the NOAEL would be replaced by an alternative, reliable parameter based on our protocols, based on the ex vivo test results, which will be combined with chemical in silico data that were presented in other speakers. All in all, I think that the concept of safe and sustainable by design could offer exciting opportunities to the growing cosmetic business, and could serve as a practical approach to enable a new cosmetic products that delivering hope for healthier life for both the people and the planet. And here is our team and our laboratory, and thank you very, very much for this opportunity.
Thank you.
Thanks a lot, Dror and Ze'evi from AHAVA Laboratories in Dead Sea area, so really hot area, I guess. Yeah, there are some questions, and also to Maria Cristina, of course, for the previous talk.
Thank you very much for an interesting presentation. I would like to ask one question. For me, it was striking that you have used sodium lauryl sulfate as a kind of a positive control, I can say, right? Because this SLS is in every product that's foaming almost that you can find in the normal shops. And it's usually stated on the first position, which means that the content of SLS is the highest in the product. I know that there are now being used alternatives as cocoa glucoside and so on, but can your approach with this new ex vivo skin model be used also as a basis for SSbD for the chemical SLS that's being used in the cosmetics?
Well, SDS or SLS are used in 2% for a long time as a golden test for irritation response, skin irritation response, especially if you want to prove that the product is good for sensitive skin. And therefore, we decided to use this one. We are aware of new proposal to replace SDS with other irritating ingredients. But thanks for this idea.
I will just add that the SLS that we put, SDS, is a leave-on. So, I mean, if you're going to put a drop of any kind of soap on your skin and leave it for several hours, you will see some irritation for sure. So this is something that you need to remind yourself as well. wish you could see
We
him.
also do have Yvonne here. She can connect you guys. Okay.
Thank you both for your very nice presentations. I would like to ask the presenters to comment a bit on the approach that you follow for this type of testing. Professor at the beginning was in vitro with cells that were first cultivated and then exposed to the shear stress. But then, for me, it's not clear if those then exhibit a phenotype that it's really related to in vivo organisms, because in vivo, they are growing while exposed to shear stress. They are not only exposed to shear stress in the latter phase of their life. So I think that could be a bit of a conservative approach. And in this case, the other presenters that were using ex vivo samples, which already have a phenotypic that can more relate to a viable organism that had these tissues before. So can you comment on this type of different approaches that you both follow? Thank you.
Yes, I agree that in the physiological condition, you continue to have your shear stress. But at least you can start evaluating if there is something similar to the static one, which are at least the mechanisms involved in this effect with chemicals and nanomaterials. And then, of course, in vivo are also needed, the test, but at least we can start in this way, avoiding in vivo analysis when not necessary. The turbulent flow, we cannot use it now because we want to see the physiological condition. Otherwise, you can also use other kind of flow, also mimicking hypertension, for example. So the pathological condition. But first, we have to know what happens in the physiological condition. But this is a first step, of course.
Okay, thanks a lot.
Right, so very short questions because I know that we have to move to the next presentation. So actually I have one question to you, Dror, and then another one to you, Maria Cristina. So maybe starting with Dror. I saw that in your presentation you mentioned, for example, exposures to up to 96 hours, right? So do you have experience like prolonging the experiment for maybe days, weeks, or even like months with these models?
Well, actually, you can keep the model for about almost 3 weeks in the lab. However, you see the reliability start to reduce significantly. However, in this work, we kind of want to see, the limited of until 4 days. Of course, it would be more interesting to see, but I guess eventually, as you proceed, you see that the levels of each kind of cytokines kind of reduce the same, like the untreated eventually. So this is what we saw, but this is something that needs to be tested and to verify that with the time, what is the levels of the cytokines after 2 weeks even.
2 weeks, very good, thank you. Maria Cristina, so a quick question. So very nice models in the microfluidic system as well. I was curious about, for example, two special things. So for example, if you measure barrier integrity and barrier permeability in your model, and a second very short question as well concerning, for example, the suspension of AdMa, advanced materials or nanomaterials. So if you have any special protocol, for example, to suspend these nanomaterials, in the continuous flow system?
Okay, really, thank you for both questions. In the first case, I start with the second one. Of course, the ULOTS University has stabilizers, suspensions, and everything set up. This is why in the static condition, we also evaluated that condition. With the flow, we are going to see what happens because the experiments are in progress, and this is another important point. The first question was related to the barrier integrity. For the barrier integrity, you need a different chamber because you need 2 chambers connected, an upper and down chamber, and then in the middle you can have your barrier, you can set up your barrier, and then make this kind of analysis. At the moment, it's only mimicking the blood flow.
Thanks a lot. One question to the colleagues from Israel and the other one to Maria in the lunch break, all right?
Okay. Thank you. I think the one question will be for both, because I just want to know how they validated their studies, since they neither used in vivo to validate if it can be used on humans or animals. They all use this in vitro, the other one ex vivo. So how did you validate your studies, or is the study still ongoing? That's my question, for both.
Please, AHAVA colleagues, you are now in the role of answering this one. Thanks.
Well, we believe that we have to go, there is a way to go to ECVAM validation, approval of the new method. We're working for it as part of the SSbD for Chem EU project. However, we have more and more results that enable this, and I think the need is very, very high, since currently new ingredients for cosmetics, it's very, very tough. I mean, the ban of animal testing for toxicity on the one hand, on the other hand, the need for new nano and non-nano ingredients to be incorporated really push, and I hope that the ECVAM will approve our proposal for this technology.
I will just add that, from the 16 years I've been working with the ex vivo skin model, we see the pattern that repeating itself every single time on every kind of different experiments. And that leads us to the thing to understand that there is something that is repeating in this model, and that can be used and exploited as a very good ex vivo skin model.
Okay, thanks a lot for the discussion. Thanks a lot for the really cool presentations on these emerging in vitro and ex vivo models for human hazard. And now we look into ecotoxicity, NAMs, provided by Alberto Katsumiti, a very international guy from GAIKER. He told me yesterday, born in Brazil, has a Japanese father, talks many languages probably, and lots of roots in his genes. So let us know more about ecotoxicity.
Thank you very much, Martin, especially for this background on my heritage, actually. So very, very good. So I think we have the slides here. So very good. So now is the transition from human health to ecotox. I don't know how many people here in the room are actually working with ecotox, but I think it would be good to just show like a little bit of the other side of what I still call like health, right? So we have human health, and we also have environmental health. So basically, this is the work that we are doing in several projects, for example, related with environmental hazard assessments, especially for the assessment of the hazard of advanced materials. So, well, as you might know, so in the context of the REACH regulation and also COP, so hazard assessment for the environmental well, for the environment is needed especially, for example, for the aquatic toxicity. So aquatic normally, for example, addresses only fresh water, but I think it's very, very important to remember that we also have the marine environment, which is another target for many, many chemicals and materials as well, and also terrestrial ecotox. So here in this slide, I'm listing basically the most used standard guidelines that are used, for example, to address especially freshwater aquatic toxicity, but also terrestrial ones. So we have many methods to address, for example, short-term toxicity using invertebrates. So we have long-term toxicity using invertebrates as well. Methods, for example, to address the effects on aquatic plants. But also we have the short-term toxicity in fish, Daphnia, microalgae, and so on. So in this list, we have here highlighted basically the methods that are based on alternative species, but also in vitro methods. And also in this second part of the slide, so basically the ones highlighted here for basically they are based on in vivo methods, so based on the organisms, so using the organisms. So saying that is basically because this talk really goes to the focus, to put a special focus on the use of the potential like new approach methodologies, but based on in vitro methods using fish cells, but not only fish cells, but also many different types of aquatic organisms, but based on cells. So Here, a very brief definition of NUMs, but I think we don't have to go through this definition because most of us here, we know what NUMs are. So basically, new approach methodologies based on in vitro, in chemical, in silico, but also in vivo methods. So the in vivo methods, of course, like more focus on the minimize the use of animal models, for example, for the research. So in these slides, I'm highlighting basically the possibility of using fish cells for these assessments. I think it's very, very important to highlight, for example, in terms of in vitro toxicology, that we have more than 4,000 cell lines established for mammalians. So most of these cell lines are used for many, many different types of in vitro studies. So studies coming from very simple assessment based on only like one monolayer of cells to more complex models using, co-culture, tree culture, tetraculture, or even more advanced models as Dror mentioned here, Maria Cristina as well with the microfluidic system. So what happened in the environmental part of this? So the environmental part of this is quite similar, but very different on the same time, because basically we have here 944 cell lines established from fish, so it's a very little amount compared to the mammalians. But if you go to the main cell banks that we have in Europe, Europe and America as well, so ATCC and DSM-CITA, we see that we have a very few cell lines coming from fish that are available for this, which for me, from my point of view, highlights the importance and really puts like an effort on, better develop these models, because they have basically the same applicability of the mammalian cells or human cells for toxicology as well. So here in this scheme, we can see a very simplified scheme showing, for example, the cells that we can obtain from a fish, for example, and the types of cell culture that we can use. So of course, we can isolate cells from gills, for example, from the intestine, from the brain, from the liver, and we can establish the cells in a primary culture, but we can also potentially immortalize the cells and use as a model for a monolayer model, for example, co-culture models, organoids, and even like more complex systems. So the potential basically of applicability of these in vitro systems are basically the same potential that we have for human health as well. So the main focus here, I think, is the applicability and the starting points of the OECD 249, test guideline 249, which is basically the only one using fish cells, the only test guideline from the OECD using fish cells, which I think is very, very important and very like a good first step on the use of these models as a standard models. So I think there is a very nice sentence in the guideline that I used to put like, here, like quoted. So 1 plate of the cell models is equivalent to 1 fish embryo test. So basically the OECD test guideline 236. And 1 acute fish toxicity test. So basically the OECD test guideline 203. So basically, after many comparability studies, they saw that the two models, the in vitro model, really aligns really, really well with the fish embryo test and also the in vivo test using fish. So this test is very simple. So it uses a cell line from the gills of rainbow trout. So the format is a 24-well plate format, which is a very simple model to work with, very simple and very easy to use model. And basically this test guideline is based on only 3 endpoints. So the 3 endpoints are based on cell viability. So basically we measure cell, lysosomal integrity, we measure metabolic activity, and we also measure integrity of the barrier, the plasma membrane barrier. So one very good aspect is with a single test, we can assess these 3 endpoints, because basically it's a combined assessment with these 3 things. So I think it's a very good in the sense that we can, with only one assay, get information about 3 different endpoints that are related with cell viability, of course, but they are different points of view. So here, on this slide, I'm basically listing, for example, the applicability not only of the OECD test guideline 249, but also like different fish cells, for example, using different protocols. So as you can see here, from a long time ago, since the 80s, for example, fish cells have been used for the assessments of the effects of metals, for example, pharmaceutical compounds, nanomaterials, and advanced materials. They are used also for long-term toxicity. So in this study, for example, the authors explored the possibility of keeping the cells for 14 days, so which in vitro is quite like a long time for exposure, and also for the predictability of in vivo effects. So normally, for example, these effects are combining not only the in vitro method, but also the possibility of using in silico. So for example, for the ones that are more familiar, so the IVIVE, the possibility of extrapolating these in vitro results to the in vivo condition. Not only the fish cells are an interesting model, but I think we have also to think about, for example, species from different environments. So here's another example of using cells from marine organisms. So here I just highlighted the use of one of my favorite animals in the marine environment, so which are basically mussels, but of course not only mussels we can use oysters and also like even fish. So here in this, very, few publications, but basically using these cell models, so in this case, for example, hemocytes, which are basically immune cells from mussels, to basically explore the toxicity of advanced materials. So in the first case, it's a polymer, so it's similar to a microplastic, but it's actually the combination of this polymer, so this plastic polymer with the silver nanoparticles inside. So as you can see in these TEM pictures, we can identify, the polymers inside of the cells. So we can go from big particles of 4.5 microns in size to 0.5. And then in the other case here, we have how we showed, like, for example, graphene, so the internalization of graphene inside of the cells. These two other studies basically on the effects on the cytoskeleton of the cells. And this is a very nice one that I used to show, which is basically the COMET assay. So I don't know if you're familiar with the genotoxicity assay, but basically here we're exposed to basically the same advanced material, so this polymer with silver, but with different functionalities. So one is a positively charged particle, the other one is a negatively charged. So we see a very clear effect of the positively charged particles here provoking basically the DNA breakage. So you can see the particles actually attached to the DNA in the assay, so very clear responses. So moving forward, because I only have like 2 minutes, so we can also explore the possibility of using, for example, earthworms cells, so primary cells coming from earthworms. So you can see many different applications of the cells. We use also for cell viability assays, but also to address, for example, mechanisms of toxicity. So many of them, so genotoxicity, oxidative stress, inflammation, and so on. Specifically, for example, the use of this guideline, 249 for advanced materials. Very complicated because we all know that how complicated it can be, like to suspend the advanced materials, for example, in the cell culture medium. So of course, we need accurate exposure methods, we need reliable testing, and we need also that these suspension methods should be very environmentally relevant. So how we can do this, considering, for example, aggregation, agglomeration, sedimentation, and flotation of these materials. So recently, we worked on an optimization of a protocol. So we started, for example, testing and studying the possibility of using the most used protocol. So using, for example, 2% of BSA and also sonication. But then we found that, of course, BSA is very relevant for human health, but not necessarily for the environment. Sonication, we found an effect, especially related with graphene, which is basically the generation of reactive oxygen species from the surface of the graphene layers. So then we end up with this optimized protocol, which is basically using natural organic matter in a very low suspension. So basically we steer for 72 hours. And then the natural organic matter forms this coating on the top of the graphene and suspends quite well the material in the system. So just a few examples of the applicability. So as you can see here, for example, the use of the test guideline using this optimized protocol. So the protocol basically works really well and allows us to study the effects of graphene in the cells. We also compare, for example, using exactly the same guideline, but changing the cell types. So basically using, instead of glial cells, we are using gonadal cells to address hepatotoxicity, or brain cells to address neurotoxicity. And then we also extended the test method from 24 hours for up to 28 days, which is a very interesting result because we can see that graphene in a very short exposure is not highly toxic, but if we extend experiments for up to 28 days, we see that the effects start to be very, very clear in the cells. So take-home messages. Fish cells, they represent good candidates as NUMs, for the HAZA assessments. So we can explore not only the glial cells, but also different cell types from gonadal tissue, brain, liver, also different fish species, not only freshwater, but also marine, different cell culture models, so co-cultures, 3D, microfluidic, so we see differences in sensibility of the cell types. We can also move from short to long-term, so to address long-term toxicity as well. And also, of course, testing advanced materials can be tricky, so we need to work on soluble chemicals and well, move from the more easy situation with soluble chemicals to AdMa, which is more complex. Very last slides, so just to show this, because I think it's very relevant, especially in the context of SSbD. If you compare, for example, again, with the same sentence that I mentioned before, like quote said, you know, that this cell culture is comparable to the fish embryo test and also comparable to the fish test. So if you compare, for example, the three tests, we see that one is like 24 hours exposure, the other one is 96 hours exposure, similar to the other one, the in vivo one. But if you compare, for example, the days of the duration of the test, and especially the cost, you know, for running this assay. So basically, these are the costs of most RTOs. So basically, did consultation with different RTOs, you know, to consult about the price of the test. We can see that actually to run OECD test guideline 249 is quite cheap, so 1,800 euros for 6 samples. And then if we compare these with the fish test, so OECD test guideline 203, this is much more expensive, so 14,000, which means that this might be a very good option, for example, for a tier 2 method, you know, for screening of compounds. So thank you very much. So this is basically the EcoTox group in GAIKER. So if you have any questions, just ask me probably in the lunchtime.
Thanks a lot. We keep the questions for the many breaks that we still have. Alberto is going to be around the entire week. He is also going to be very active in the trainings. You will have lots of interaction time to discuss with him. Thanks for this overview. And now we switch to the in silico methodologies. They are also new approach methodologies. We have seen ecotoxicity, we have seen human hazard models on in vitro and in ex vivo systems. Of course, there are many, many more on this Resilience 21 cluster projects. Many things are developed currently. And now we have the pleasure to listen to Anita Sosnowska and maybe also Natalia Butawska are going to be coming up from the QSAR Lab in Gdansk, Poland, being involved in many EU projects. Let me pull it up. And also Anita and Natalia are going to be helping us with the training tomorrow. All right.
Okay, thank you very much for introduction. I would like to talk about computational models and how they can support SSbD approach. So we know from previous presentation that SSbD incorporate safety, environmental sustainability, and functionality early in the innovation and development of chemicals and materials. And in this way, in silico methods, computational approaches such as QSAR, read-across, molecular docking, and machine learning algorithm can help in early prediction of hazardous properties and also environmental fate of substances. These methods facilitate data-driven prioritization and screening of chemicals candidates, also reducing time and resources, and also need for animal testing. They align with regulatory frameworks and are integrated into new approach methodologies used for chemical risk assessment. What is QSAR? QSAR is a quantitative structure-activity relationship modeling, and for the development of QSAR models, we need a set of chemicals which are similar to each other. We have to numerically describe their structure. And for this set of compounds, you also need experimental values for activity or physicochemical properties that we would like to model. Then we combine those two sets, and applying different machine learning methods, at the end we derive the mathematical formula that can be applied for, that relates the structure with the activity. And also this mathematical equation could be used further for prediction of the modeled endpoint for new compounds for which we don't have data. So for what purpose do we use QSAR? First for predicting missing values, for data gap filling, but also for searching structural features that affect model activity, so for deriving the mechanistic interpretation, structural alerts that are really important in mechanism. There is a wide range of applicability of QSAR methods. They can be used for virtual screening of compounds, for assessing mobility, fate, toxicity, also in green chemistry for searching compounds with desired properties, but also safe for the environment. They can also be used in designing new materials, including innovative advanced materials. We have two kinds of application of QSAR models. First, we can develop a model if we would like to know, for example, the mechanism of toxicity. We can develop a model for our compounds and see what is important then. But if we would like only to predict missing values for target compounds, we can apply existing ones. Each model should follow established OECD principles. They were published in 2004. There are 5 golden principles for validation of QSAR models. There are, as I said, 5 rules. First is a defined endpoint, so we have to know exactly what kind of toxicity was measured, and the data for training the models have to come from the reliable sources. The second one is about a clear algorithm. It means that each step that have been taken to develop QSAR model have to be described in such a way that it could be easily recreated in any time. Then we have to define the applicability domain of the model, so the space in which model works properly, in which structures we will get reliable results. We have to calculate and interpret the different parameters, and at the end, we have to indicate the mechanistic interpretation. In 2023, the OECD released the updated version of those rules. This is QSAR assessment framework, and those rules mainly contain the principles for assessment of QSAR predictions. They are not concerned of the developed model, but they rely on the predictions and the results of different kind of models. What is very important nowadays, QSAR has to be in line with FAIR principles, so it means that models should have to be findable. Models and data sets should have unique persistence identifiers. Data for training the models and also all metadata should be indexed in repositories. Models also should be accessible, so it means that the licensing should be clear, and whenever it's possible, those models should be openly available. In case of interoperability, models, so the data used for the models should be formatted in standardized vocabularies and ontologies. And in case of reusability, there is need for full documentation, so it should follow also this OECD principle. So we have to have information about the algorithm, applicability domain, and validations, and all parameters. Where can we find QSAR models? First, of course, in the literature, there are many papers concerning QSAR and QSPR models and other predictive approaches. But if you would like to apply such a model, you have to remember that first you have to evaluate such a model if it fulfills all OECD principles. But also what is very important, you need to perform a critical evaluation of the data that were used for training the model. If the data came from reliable sources, and if your target compound, your compound for which you would like to predict some endpoints is similar enough to the training set. Then what you have to do is to calculate molecular descriptors, to recreate the model and perform the predictions. In those 3 stages, you need some expertise in machine learning. And then you have to assess if predictions are reliable and the results are okay. There are also other possibilities of finding QSAR models. There are open source tools which implement models for different endpoints, for physicochemical properties of compounds, for toxicity, ecotoxicity also. Everyone can use those tools. And if you would like to use those tools, you have to first select the proper models. So you need to think, so you need to evaluate if the property or toxicity which you would like to predict is really this toxicity which was modeled in the approach. Then you have to introduce the input to the tool, and the tool itself perform the predictions. And what you need to do at the end, it is a critical expert assessment. So here you need a knowledge what is important in such an assessment. We will do it tomorrow in a hands-on session. There is also other possibility. You can use a platform like LASA or Instem. They are not free. They are working more in a collaborative way between clients and themselves. And here, what is the advantage of those platforms? At the end, at the critical expert assessment, you have also help from the platforms, and they will assess the results together with you. Also, in the QSAR Lab, we developed tools that can be used for predicting different endpoints. First one is in silico lab, which implements various models, now mainly for neurotoxicity, but also for other properties of compounds. Also for ionic liquids, it is possible to predict different endpoints using in silico lab. And the other tool is MQTB. This is a web-based tool which implements different machine learning algorithms to predict genotoxicity and mutagenicity for nanomaterials. And to sum up how the in silico approaches could be used in SSbD framework. So first, in early stage, in hazard screening, so for identification of potential hazards associated with new chemicals, for prioritization of safer chemicals. We can use those methods for data gap filling in early step of development, where the data scarcity is of course common. They are integrated with regulatory frameworks, and they are recommended for screening and for risk assessment. And also they support a tiered approach, mainly the tier 1 and 2, using in silico methods, you can prioritize substances. And what is also very important, they providing a continuous feedback on hazard and sustainability profiles of the chemicals. Yeah, and the case studies, how they can be used, this will be the presentation of Natalia. So welcome everyone, and after that methodology introduction done by Anita, I would like to show you some case studies, some practical example how to use the QSAR approach, also the QSPR models and another in silico methods to predict some properties, but also the biological activity of the chemical compounds. And the first case study is about the searching for chemicals with desired properties. And here we're dealing with the ionic liquids. The ionic liquids is really interesting chemicals because of their toxicity, but also the specific physicochemical properties such as low melting point, the solvatation rate, also the marginal vapor pressure, and so on. But not only the physicochemical properties of the ionic compounds is important, but the toxicity and ecotoxicity, the researchers put so much interest in that case. Normally, the toxicity or ecotoxicity of compounds is considered as unfavorable feature, but ionic liquids could be developed and projected in that way to keep the toxicity in desired level. And this gives us the opportunity to design the new biocides. And the first example I want to show you, it was split to 4 different phases. And now, step by step, I will give you some introduction to every phase. The first phase was about the generation of theoretical ionic liquids, and here we had more than 700 structure of cations and 80 structure of ions, and then mix each with different combination. We can generate the whole list of more than 60,000 ionic liquids to be further analyzed. So after this step, the structure of the ionic liquids were designed, and the descriptors were calculated to then screening using the QSAR models. And the second phase, it was about developing the QSAR models. And here, to develop the QSAR models, we need the list of the experimental values for our toxicity, our endpoints. And in this example, our endpoint was the toxicity of ionic liquids against Staphylococcus aureus. Of course, the collected data from the literature should have the same, should be measured under the same condition, exactly the same condition, to be the one data set to develop the QSAR models. In this case, we had the binary responses like active and inactive compounds, so that's why we can build the classification tree model. And in the next phase, we can use that classification model to predict the activity of previously designed theoretical set of ionic liquids. And using our QSAR models, we can say that only 25, so less than the half of the whole list of compounds, was predicted as active and was in the applicability domain of the model. The next step, the environmental mobility, give us an opportunity to, again, reduce whole set of compounds. How? We use the aquaboxio tool to check the chemical in the water, sediment, organic matter, using the 5 implemented QSPR models, which can predict the water partitions, n-octanol water partition coefficient, solubility, and so on. And after implementing that 5 QSPR models, we can set that only 1,000 of ionic liquids in a domain of that whole set of the models. The last phase about the environmental sustainability, it was also to classify the substances using the definition of the OECD test. So the substance which go to biodegrade less than in 60%, we can set that is hard to biodegrade. So after that step, we can set that only 70 ionic liquids have a good property and also are environmental friendly. And here, this week's slides to conclude how it's gone. So we started from more than 60,000 of ionic liquids. Then we can reduce it more than half. Then we reduced again it 65 times. And at the last step, we have only small set of ionic liquids with desired property, which is toxicity against Staphylococcus aureus, and it's also environmentally friendly. So that small set of compounds, it could be next experimentally validated. The second case study I want to present you is about the theoretical design and evaluation of functionally improved salicylates, because the salicylic esters used as the ultraviolet absorbers in the sunscreen, it's working good, but also have been proven that can cause the skin sensitization of estrogenic risk. So the main goal of this study, it was to design some substitutes of salicylic esters, which has the good UV absorbance behavior, but also the minimalize the estrogenic effects. So for this purpose, the researchers collect the data, they make the data sets of the 73 different potential compounds that could be new substitutes for salicylates. And to do that, they use molecular docking, and only another tool, as a initially assessment of the property, and then to use the 2- and 3-dimensional quantitative structure activity relationship model to predict the estrogenic effects, but also the UV absorbance. So to get to know how the chemicals could be bind to estrogenic receptor, they started from the molecular docking. And then the molecular docking score was used as a measure to set how the strength is strong between the chemical and between the receptor. After that, they construct the models, a lot of models, and within it, based on the best predictivity, they use the final models for the both properties, the UV absorption, UV light absorption, and also the estrogenic risk of the chemicals, and then they screen the whole candidates, whole group, from the beginning, the 73 compounds, to check which one have the best properties and activity. To say something more about the QSAR model, how to build it, and how that physicochemical, that structure feature, it's connected with physicochemical and property and biological activity. So here we have the two models. The first one, which was developed for estrogenic effect models. And in this case, we had two key features. The first one represents the covalent hydrogen bond acidicity, when the second one reflects the hydrogen bond basicity. And that both key descriptors in the model could help us to say how the structure, how the chemicals can bind with the hydrogen bonding, which highly related with their possibility to bind to the estrogen receptor. The second three-dimensional QSAR models were developed using only one descriptors. It was the HOMO eigenvalue for the UV absorbance model. And here we can say that the HOMO eigenvalue, so the highest occupied molecular orbital, its energy, it's linked closely with the ability of chemicals to absorb the ultraviolet light. So that's why it was used as a crucial indicator of the prediction of UV light ability. And after implementing that two QSAR models, the researchers can take into account two compounds finally, which have the low skin sensitization, low estrogenic risk, but also have high UV absorbance abilities, low skin permeability, and also do not cause skin irritation and eye irritation. So coming up to the conclusion of this example, we can say that UV absorbance of salicylic esters is driven by efficient electron migration between the orbitals and also driven by the strong vibronic interactions. And the last case study, I will try to be shorter, it's about the possibility of design the new lubricants, again using the theoretical approaches, the in silico methodologies, to check and to avoid the too broad experimental testing set. So starting from some conclusions that our researchers developed only 3 ionic liquids as a candidates to supporting to improve the automotive lubricants. But how they start all that study. So they using the QSPR methodologies to rapid and also reliable prediction of the chemical properties, physicochemical properties of chemicals, to narrow down in the first step the whole list of the candidates. Then the most promising ones, we understand about that one which has the lowest kinematic viscosity and also are designed in the set of ionic liquids, are going to the molecular dynamics simulation to check the chemical feasibility. And the next and the last step of that research, it was to molecular dynamics simulation used to proper validation of the critical lubricant properties. So after the synthesis of chemicals, go to the laboratory, undergo to the testing and check the chemical feasibility and also to possibility of the manufacture in the lab scale. Also, the thermodynamic feasibility was checked using the theoretical methods. And to conclude that using the computer-aided molecular design could be also combining with the molecular dynamics could be really good strategy to narrowing down the set of compounds and also to check only few candidates to the further experimental testing. And as I said, it was my last example. Well, thank you very much for your attention.